everybody. So today we are going to read chapter number 2, Biological Classification of class 11th. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for their own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological character to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which have red blood and those that did not. In Linnaeus' time, two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all the plants and animals respectively. This system did not distinguish between the eukaryotic and prokaryotic, unicellular and multicellular organism, and photosynthetic green algae and non-photosynthetic fungi organism. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, mode of reproduction, evolutionary relationship, etc. Classification systems for a living organisms have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all the different systems, the understanding of what groups / organism be included under these kingdom have been changing. The number and nature of other have been understood differently by different scientists over the time. R.H. Whittaker in 1969 proposed 5-kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The criteria for classification used by him include cell, structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. The three-domain system have also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a 6-kingdom classification. You will learn about this system in detail at higher classes. Let us look at this 5-kingdom classification to understand the issue and consideration but influenced the classification system. Earlier classification system that includes bacteria, blue-green algae, fungi, mosses, fern, gymnosperm and the angiosperm under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, which is cyanobacteria, with other group which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under fungi. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plant though they were also showed a characteristic difference in their cell wall composition. The fungi had chitin in their cell wall, while the green plant had the cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, that is, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in the kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella. Chlorella was earlier placed in alga within plant and both having cell wall, with paramecium and amoeba. Amoeba lacks Amoeba, which were earlier placed in the animal kingdom, which lacks cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification has changed. The kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt have been made to evolve a classification system which reflects not only the morphological, physiological and reproductive similarities but also phylogenetic. In this chapter, we will study characteristics of kingdom Monera, Protista and fungi of the vertical system of classification. The plantae and the animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in chapter 3 and 4. So, number 1, kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans where very few other life forms can survive. Many of them in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape. The spiral coccus, plural is cocci, the rod-shaped bacillus, plural is bacilli, the comma-shaped vibrium, plural is vibrio, the spiral-shaped spirilla. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophic, that is, they depend on the organisms or the dead organic matter for food. So, the next topic is archaebacteria. Archaebacteria. These bacteria are the special since they live in some of the most harsh habitat such as extreme salty areas, halophiles, hot springs, thermoacidophils and marshy area, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of the several ruminant animals such as cow, buffalo, and they are responsible for the production of methane, biogas from the dung of the animals.